Welcome to How Do We Make Genomics Everybody's Business? I'm Sarah Norcross and I'm the Director of the Progress Educational Trust, a charity that was founded back in 1992, before quite a few of the audience were born, I think, looking at you, to improve the public understanding of science, law and ethics in the fields of human genetics and assisted reproduction, and also embryology. And what PET aims to do is to improve choices for people affected by genetic conditions and or by infertility. We do this by aiming to promote the responsible application of science, and we engage with the public about these issues and we lead the debate. We identify the challenging topics, encouraging meaningful public discussion in this area. We influence policy so that it's better aligned with science and so that people can access these treatments in a timely manner, and informing and explaining. And we do this through holding events like this and also by publishing accurate and unbiased news in our online publication BioNews. And we're delighted to be working with Genomics England as part of their genomics conversation. So why are we doing an event about this? Well, that is because the Chief Medical Officer, Professor Dame Sally Davis, argued in her report, Generation Genome, that the key to advancing genomic medicine will be helping patients to understand that by agreeing to use data about their illness, they bring direct benefits to themselves. And she went on to say, we need to continue and strengthen an open dialogue and make sure that the argument for joining in is heard. To achieve this, we need to maintain patience and public trust and make genomics everybody's business. But business at this event has a broader meaning because NHS genomics is a key focus of the UK's current industrial strategy and life sciences sector deal. How do the public and private sectors work together on UK genomics? So these are really big and important questions for the UK. So we're delighted to be in Manchester for this event because it's a fantastic city. I was born here, so I'm very biased. And also because, we're here, it's Expo, it's the Northern Powerhouse, there's the Manchester Centre for Genomic Medicine, which is part of the 100,000 Genomes Project. And also, speaking as a Mancunian, I can say that Mancunians love to know everybody's business, and most will happily tell you your business. So on your way home, don't be surprised if you're standing at a tram stop and someone starts to talk to you. It's quite normal here. And at 9 o'clock tonight, the cast of Corrie, that's Coronation Street for non-watchers, will be sharing their DNA secrets on TV. Who knew? I'm quite intrigued to find out what those are, and I think quite brave of them to do it. So I just need to now introduce your chair for this evening, who is Professor John Mattick. Now, John is the Chief Executive of the Genomics England. He's joined the team coming from Sydney in Australia. And I wanted to give him a warm Mancunian welcome and also to say that I did look, because we're obsessed with the weather in Manchester, that it does actually rain more in Sydney than it does in Manchester. So that's, just to put that out there. But about John, he's, got an international reputation as a scientist and has won many, many awards. And in the media, he's been called a pioneer, a soothsayer, and Pied Piper, and has described it being at the vanguard of transforming clinical care through genomics. So, and he's a precision medicine evangelist. So he's, really passionate about placing diagnostic and curative powers into doctors' hands to getting this technology to patients. So please give a really warm welcome to John.
Well, thank you, Sarah, very much. And it's just delightful to be here. It's my first time in Manchester, I regret to say, but it won't be the last. And I have to respond to your comment about Sydney being wetter than Manchester. It is true. We have 1.25, they have, I should say, 1.25 metres of rain a year. But the other fact is that we have only 30 cloudy days a year. So when it rains, it pours, which is a nice way to do it. Look, it's great to be here. I just want to explain, first of all, the format for this evening. There's going to be 4 presentations of 10 minutes each. We'll hold questions until the end, and then we'll invite all of the 4 speakers up to the front to take questions. I'll put some general questions first, and then I'll invite questions from the audience, and we can open it up for general discussion. The only thing I would say to get the ball rolling, because we have 4 very distinguished speakers to follow, is that, and as Sarah says, I guess I'm an evangelist, a missionary perhaps, you know. I've been in the business for quite a while, studying the human genome, which I think is the most extraordinary suite of information that exists on this planet. I think I'm right in saying that its information content is about the same as the full version of Microsoft Word, but it produces something that walks and talks, which is extraordinary. And we don't understand it very well. We understand the protein coding portions of it pretty well, and they're the things that get damaged in the so-called rare diseases, which are not rare at all in aggregate. But the rest of the genome is where the information that makes us individuals lies, and where all of the variation that underpins our susceptibility to complex diseases lies. So we need to actually understand and have at our fingertips the whole genome. And what is going to happen over the next few years, I think, is a complete transformation, not only in healthcare, but also in medical research and the way we go about understanding human biology. Because every feature of our biology is ultimately either completely or almost completely or strongly influenced by genetics. I point out things that perhaps might not be so obvious to you, but facial features, essentially 100% genetic. That's why we look like a mixture of mum and dad, and we know it's 100% genetic or close to it because identical twins are basically phenocopies of each other. So if you run the same program twice, you get the same outcome physically. That's the 3 million differences between us in our genomes are what make us what we are as individuals. Our susceptibility to infectious disease is almost entirely determined by our genome. Beautiful work in New York by Jean-Laurent Casanova has shown that if you give a group of people, or mice as the case may be, a standard dose of tuberculosis or a virus of some sort, that some will get the infection and some won't. But it's not a random subset because of idiosyncrasies in our immune system. We can already, to some extent, although we don't do so yet, predict which children will have adverse reactions to vaccinations, which will help solve that conundrum in the community much better, because those are the kids that have immunodeficiencies and we can pick up by genome sequencing. Essentially every feature of our biology is determined by our genome. And we now have the opportunity to look under the hood at that information. And I'm imagining, not just in the clinic, but it is ready for the clinic, and particularly as the costs come down, but in understanding human biology, soon we'll have millions, literally, of high-resolution genetic information on citizens, privacy protected, that's a very important feature of this, but with accompanying clinical records and very rich data, which I think is not yet fully appreciated in its impact, from the smart devices, the wearables, the implantables that are being developed, real-time physiological information. When you put those two things together, we are actually going to have the research ecology to die for, because finally we'll be studying the human and the human variation in all of its magnificent dimensions. So this will transform the way medical research is done. Every time we find a connection between a genomic variation and something to do with human biology, where that's relevant, that can be fed back into clinical pathways. And so research and the clinic are collapsing into the same endeavour. And this is actually a challenge to both the traditional research endeavour, as we've constructed it, which tends to be reductionist in molecular biology, and the traditional clinical endeavour, which tends to be service to the point of crisis, as opposed to actually understanding our risk for cancer or complex diseases like cardiovascular disease, and intersecting early at those people who are at really serious risk to head that off by prescription of statins or by regular screening of people at high risk for cancer. And in fact, we've just published with the National Cancer Institute in the United States that using genomic information, we could predict with higher accuracy than any other test who not only is at risk for cancer, but who has cancer. So we gave these individuals whole body MRIs because, with the exception of BRCA and Lynch syndrome, colon cancer, the other cancer-causing genes are tissue agnostic. And when we did that, based on mutations that these individuals had inherited, 10% of them already had pre-symptomatic cancers they were not aware of. One poor chap had 2. But at this point, they're very easily to remove by surgery before they've metastasized, before overt symptoms come. So I think we're going to move towards a much better understanding of our own biology, what makes us individuals, and a much better health system, more efficient, that actually anticipates the fragilities all of us have in one way or another, and works to make sure that the consequences of those fragilities are mitigated or prevented by early action. So that's where we're going. I will now leave it to the others to fill this out. We've got 4 speakers. I'll introduce them as we go. Our first is Dame Sue Hill, who's the Chief Scientific Officer of NHS England and Senior Responsible Officer for NHS Genomics. Dame Sue Hill.
What I'm going to be talking about really is setting the context in terms of the NHS and the NHS services that we're about to put in place, but the history that we're building upon, and why, as we move forward, genomics will be everyone's business, whether it's clinicians in the NHS, whether it's patients, or whether it's people in our general population. So the NHS will be the context, and hopefully we can come back a bit later on and have the conversation about what else we need to do to make sure everybody's aware of what we're doing. So the first thing is the NHS has been a world leader in clinical applications of genomics. Both before the NHS actually came into being, the first genetic counselling clinic was held at Great Ormond Street in 1946. The first consultant clinical geneticist was appointed in 1958. And so there's a long history in the NHS of applying genomics for clinical care purposes. And really, since the NHS has had a set of clinical genetic or genomic laboratories that came into being in the 1960s, these laboratories have really taken all of the cutting-edge technologies and introduced them into healthcare. But the point that John Mattick was making in his introduction is where we are today with the technology means that we can make a step change in terms of how we look at all of those letters that make up our the whole human genome, and what that might mean in terms of our health. Both in terms of the disease or the symptoms we may be presenting with, or in terms of the ability to predict disease that may or may not be starting to be presented. And so, in terms of the NHS's capability and capacity, they've been doing this for a long time, and more recently they've used whole genome sequencing for clinical care purposes as part of their contribution to the 100,000 Genomes Project. So to sequencing whole genomes in patients with rare disease and their family members, and also in patients with cancer. So whole genome sequencing is the latest addition to that. What we've had for a long time is the government's recognition of the role that genomics can play, and long-term investment because of that, that's helped to make us a world leader, not only from a research perspective, but from the NHS perspective. And what we know as we start to plan for the NHS over the next 10 years, in terms of the NHS 10-year plan, which was set out the ambition for the NHS over the next 10 years. But associated with that, those improvements will be what we'll do over the next 5 years as part of that £20 billion that Theresa May, the Prime Minister, announced in June. So this is a very good time for genomics and a very good time for us to understand what the value will be to our patients and our population. What we've seen through the 100,000 Genomes Project and the use of whole genome sequencing is a proof of concept has been demonstrated within the NHS. So the use of that technology within routine care has been established. We've looked at the feasibility of using it and how it might be affordable in comparison to the technology we use today. We've actually entered into 2 new consent models where Participants have been consenting both for their data to be used for clinical care purposes, but also in a de-identified way for research and for discovery through industry partnerships. We've been able to capture all the data that's really important, as John has said in his introduction, to really understand this complex genetic information in the context of what the individual might be experiencing that time, or other different functional abnormalities they may have, for example. We've been able to standardise processes and pathways. We've got the beginnings of system-wide involvement to date. We've got over 85 NHS trust organisations across England involved in this project. We've got the important clinical leadership in place, and we've started to align the system alongside this. So it really started through a proof of concept project to tell us what we needed to do to comprehensively implement genomic medicine at the scale of 55 million people in our population, and the population that is served by the NHS in England today. And this is importantly being coordinated through 13 NHS genomic medicine centres. And here in the northwest we have one side of the M62 in Liverpool and the other side here in Manchester. Why this has been important is what's been a key component of this is engaging with professionals and with patients across each of those 13 geographical areas in England. So it started the genomics conversation, but it's also started our understanding of what we need to do to make genomics everyone's business. So as we move forward into October of this year, in NHS England, we'll be introducing a new NHS genomic medicine service, building on where we've been, that history over the last 60 years, to provide a more consistent and equitable offer to the patients in the population. The services within this National Genomic Medicine Service were delivered to common national standards, specifications, and also protocols. So we start to standardise what people receive, whether they're in Newcastle or whether they're right in the depths of Cornwall. Integral to the delivery of this new service will be a single and one national genomic test directory, which will set out what tests will be available and by what technology and how that will be provided and for what conditions across the country. But this will cover, for the very first time in the NHS, from looking at single genes, so single collections really of DNA, right through to the whole genome sequencing. So as we start the new service, we'll be introducing whole genome sequencing for routine care. And the only health system in the world, I think, who will be doing this in a systematic way. All patients will be given the opportunity to participate in research, and they will be asked that question when they're being offered their genomic test for routine clinical care purposes. And that will benefit them as individuals, but also benefit in terms of the learnings that will go back in and inform the potential for their care and for the care for others with similar diseases. And we will build, together with Genomics England, a national genomic knowledge base, and that will inform the work that will be done by researchers and by industry collaborators, so that we create an ecosystem for patient benefit, so we continually learn and patients continually benefit. And that will include in terms of enhanced clinical trial recruitment. So this is what the service looks like in terms of what NHS England will be introducing from October. There will be a national network of genomic laboratories delivering a national genomic testing service, set out in a national test directory. That is what NHS England will pay for in terms of the tests that will be available across England, particularly focused on rare and inherited disease, in cancer, but increasingly in more common diseases. We would retain our genomic medicine centres, and we'll be restructuring and aligning some of our other services so that we have a coordinated approach across the whole of the country in England. With Genomics England, we will be working in partnership to secure a whole genome sequencing provision for the NHS, to have the latest data analytics built upon the experiences that John and also they have already in Genomics England can bring, or others across the world. And it will be underpinned by an informatics system that enables data to flow. But importantly, through this new genomic medicine service, we'll be driving the industry, academic and international partnerships that are going to be important to make this work for the future. So this is about bringing everything together, genomic laboratories, cancer services, clinical genetic services, NHS genomic medicine services, and bringing it all together to drive seamless care, but recognising we need to do a lot of work with our workforce. This next slide just summarises the workforce developments that have happened as a part of the genomics education programme that Health Education England have been delivering, looking at the type of workforce interventions that are necessary, both for the whole workforce, for those workforces whereby we'll be rolling out services within those clinical specialties, but also for the highly specialised services. And so it's an educational continuum, and what that includes awareness raising, and particularly how we develop the primary care workforce. And this will be evolved to support our future service. And when we think about the workforce we need for the future, with the way genomics is taking us, we'll need a workforce that's planned and developed for the future service, that's both agile and responsive to changes and can handle large amounts of data. And that probably is not just about genomics. Critical to this will be how we bring together networks and how we bring together people to work as one national service to accelerate ideas, to accelerate the broader conversation, including, as you can see, through patient and public involvement. And this will be central to what we're doing. And the final slide of mine today, Sarah, is about how we take this forward. Has to be about how we work together in partnership, whether it's professional partnerships to drive the changes that are going to be necessary to embed genomics medicine, whether it's very specifically about disease areas and how, for example, we can bring better genetic testing available to reduce sudden cardiac deaths in the population, whether it's about discovery and working with Genomics England and our academic health science networks, whether it's about our new consent model or other parts of the service, how we think about the emergent technologies and they come into in the NHS rapidly within this new system, or how we get engagement at a very local level to drive new models of care and new ways of delivering that care for patients. So I hope we can have a conversation a bit later on about what we've done, what else you feel we might need to do, but actually recognising we've thought about all of these components and have the infrastructure to deliver. So thank you very much.
Thank you, Sue. It's a fantastic overview and just reminds us all that the patient is at the centre of all this, and what Sue and the NHS are building is a far more precise and personal framework for treating patients in the future. Our next speaker is Professor Nalin Thakkar, who's a consultant histopathologist at the Royal Manchester Infirmary, and more importantly in this context, I guess, is Professor of Molecular Pathology and Genetics at the University of Manchester. Nalin, please.
Good evening, everyone. Welcome to Manchester. I'm a local lad. I've been here 40 years. I came here as a student and I stayed here because it is such a wonderful city. So, and welcome, John, particularly. And you're welcome again to come to Manchester anytime you like. I'm going to take a very different angle on this. Sarah asked me to talk about research perspective on this. And I have interpreted it as, how do we enable genomics research? I hope that's what you wanted me to talk about, but that's what I'm going to talk about anyway. In terms of enabling genomics research, if we look at the sort of folks who are involved in genomics research at the moment, we would brand them as experts, generally, people who know what they're doing. If you want to expand the scope of this, you've got to bring in new researchers into this. And you've got to have the right framework for them to be easily able to slot into this. So that's what I'm going to talk about. I hope I got the message right, and I'm not talking about the wrong sort of arc here. And we'll start with, I think there are 4 critical things that we need if we want to sort of enable genomics research. The first thing is that you really need a framework that guides us on issues like consent, on ethics, what is acceptable, what isn't, standards in sample access, sample management, and what sort of things we can use them for. So if you like, this is the sort of defines the sort of basic values that we subscribe to and standards that we all accept as reasonable and follow. Some of these could be embedded in statute, some of them can be embedded in regulatory processes, some of them will be embedded in best practice, not in any other sort of binding rules. But you do need those so that anybody can engage with that research and that you're not reinventing the wheel every time somebody wants to do this. Let me give you, this is really important. Let me give you an example of when I've applied to do a genetic study in Scotland many, many years ago. And I had to actually apply to 28 different ethics committees. So I had to fill in 28 different ethics forms. And each ethics committee had a different view on what I was doing. So what you need is some sort of standardization if you really want to enable this. You also need necessary research infrastructure and processes that will facilitate this and support the research and are compliant with the standards that we've just said. So we have the ability to do the research that we want. The third thing, which I guess Vivienne will talk about in terms of, is our knowledge base and needing a well-informed researchers, research ethics committees, regulators, and research participants. When I say research participants, I mean everybody, the public, because we are all potentially these research participants when we're talking about genomics research. We all stand to benefit from this sort of research. And we're not really just talking about understanding of the science here. We really need a better understanding of methodology, quality standards, and ethical issues in this arena. And these are constantly evolving. They're not stable. So what we considered unacceptable maybe 20, 25 years ago may be now acceptable. And I'll give you an example of generic consent. If you told me 25, 30 years ago that generic consent was acceptable to most people, you would have been laughed out of the ethics committee because it just wasn't the done thing. And now we accept that as a basic way to operate. So the ethical standards are constantly evolving, and we do need to educate folks on that. The other thing people need to understand is what is the clinical impact of what we're doing. Good example of this is you get basic researchers who, without much clinical input, because this technology is accessible to everybody, anybody can do genomics research nowadays, and I proved the point by calling up the Genome Beijing Center and saying, I've got 40 samples in the fridge, would you be able to analyze them? And they said, yes, of course, just send them over, no questions asked. So there is no limit for anybody to sort of do this sort of work, but you really need to then say, well, do you understand what you're doing, the impact of the information that you're pulling out? So we've had researchers who've said that in the ethics committees where they've said that what will you do with the information? Let's say you find an incidental finding. Well, we'll send the whole genome data to the GP. And really? You're going to send all that raw data to the GP to deal with. So really, a better understanding of how you're going to handle this information, what the clinical impact is, and so on. And finally, you need what Dame Sue Hill talked about, is a clinical service that will not only handle the sort of findings from individual research studies, but the sort of for wider clinical benefit realization. And if you don't have that, there is little point doing the research. So you really need to be able to engage what you find into the clinical service, translate that into clinical service. In terms of the first two of these, the framework to guide consent and so on, and the necessary research infrastructure, we're actually in this country in a pretty good place. Slightly fortuitously, because at the turn of century we had organ retention issues that resulted in introduction of legislation to regulate human tissues. So we have existing human tissue statutory and regulatory framework which applies to DNA analysis as well. So this is the Human Tissue Act in 2004. It's in fact exactly 12 years and 5 days since it came into force. And following this, there was a research ethics framework introduced to sort of say within the law tells us what is basically acceptable to what we can and cannot do, but within this, how do we apply our understanding of how we can use the human tissues for research and in biobanking. We have very favorable regulators and a regulatory environment in this country. The HTA and HRA, I refer to them as cuddly regulators. They are really very good. They engage very positively. I know the amount of engagement that went on with Genomics England when the first ethics review of the 100K project was going through, because I was involved in that. And then also more recently when Genomics England was trying to sort of issue guidelines on collection of frozen tissues as part of normal clinical practice. Again, there was quite a lot of engagement with both the regulators, and they are really very willing to engage and operating within the rules, facilitate the research. So I think we're quite fortunate in that. As part and parcel of the sort of changes that came in about 12 years ago, we have actually addressed quite a lot of the issues that might stop us or might sort of hinder us in progress. And so we've, as I've mentioned, the issue about generic consent and consent for DNA analysis and things have been assessed, consent for commercial use. We know we can use tiered consent. We know we can advise on how to sort of use different types of consent to sort of facilitate the work. We've got guidance on removal, use, storage, and disposal of tissues and DNA. We're a long way down the road of knowing how we can protect the information for patients, and there is good governance in this area. We have got researchers, because this has been enforced for about 12 years, we've got researchers, research ethics committees, and research organizations which are also familiar with this framework. So in terms of implementing genomics research in this country, we've got a really strong foundation. In addition, research organizations have already built up biobanking capacity and infrastructure. One thing the Act did was impose standards on how we store tissues. And by driving up standards, we've actually built up capacity to biobank properly. So you'll find that most research organizations now have capacity to biobank properly. So, and I'm talking across the board here in NHS, universities and industry. So there is better handling of and storage of tissues, which gives us better quality of tissues, which are essential for genomics research. We have got improving quality of data associated with tissues. Tissues are nothing without data. They don't mean anything. There is accountability and good governance. And as I said, there is a facilitatory regulatory environment. So an example of this is that biobanks in this country can have generic ethics approval. That means anybody who wants to obtain tissues from from bank with such an approval doesn't need an ethics approval themselves. So you can have multiple research projects operating off a single ethics approval. So we operate in a very facilitatory regulatory environment. And we are getting better at wider access to tissues with biobanking. Of course, in the catchment area of biobanks, you get better accessibility of tissues, but we have also got national initiatives like the UK CRC Tissue Directory, UK Biobank, disease-specific research tissue banks, and so on. And finally, we have registries which are disease-specific or just for rare diseases. So in terms of the first two, I would say we have got a really strong foundation of where we are. In terms of the next two, in terms of knowledge base and having well-informed researchers, RACs and regulators, research participants, I think we need to do a lot more, and Vivienne, I suspect, will talk a bit more about that. And in terms of the clinical and clinical laboratory services, Sue has addressed that issue. So I think I will stop there because I have run out of time. I've got a few more slides, but I will stop. Thank you.
Thank you, Nalin. It's a very good overview of the structure to enable this, us to be able to study the wonderful dimensions of human biology. And we will talk more about it. Our next speaker is Paul Matthews, who is the Head of Strategic Partnerships at Global Gene Corporation. Paul, welcome.
Thank you to the Trust and also to Genomics England for the invitation to come and speak to you, and thank you for actually being here on such a nice non-rainy day in Manchester. We're talking about, I was born in Manchester, although I have lived down in the south for far too long and I've lost most of my accent, but it's good to be back up here. So I represent Global Gene Corp, and I've been asked to talk about the commercialization side of genomics. And so very quickly, as a company, what Global Gene Corp are about, we're about democratizing genomics to positively disrupt health outcomes for everyone. But what does that actually mean? What are we doing? There's a number of things that our company have been working on since it was first founded in 2013 by two clinical geneticists, one working on the east coast of Boston, one working in Singapore, who were very frustrated that the promise of the human genome hadn't delivered in the way we thought it would do for medicine. And I worked on the Human Genome Project way back when it first started, so I'm coming at this from that side. They worked from the clinical side, and one of the things that they realized was that the genomic data that we have available to us in the world, 81% of it is a European ancestry, so a Caucasian data set, if you like. 19% is non-European, and a lot of that is locked away where it can't be accessed by anybody to use because it's Chinese data and we can't get hold of it. So the company set about addressing the issue of a lack of diversity in the data, and that's led to misdiagnosis and misinterpretation. So we have set up in India, India's largest biobank, and recently the largest sequencing program for the Indian population. And in order to collect data from India, we have a number of different technologies that we use to gather phenotypic information, clinical samples from patients in the biobank. And then our business model is collaborating and working with drug discovery, pharmaceutical companies, using that data along with the European data sets to speed up drug discovery, biomarker identification. And while we've been doing this, there's a lot of other things that have come into play as well for our company. So gathering information from the 29 states in India it's not an easy thing to do. So we've developed mobile technologies to gather the phenotypic information. We don't have an NHS service in India. So we have people going around in real time adding high quality clinical information about our patients. We've also developed on the far side over there something called a GGC-Me app, which is a way of connecting an individual's genomic sequence with the individual. And I'll talk a bit more about that in a moment. But what it facilitates is really a creation of a precision medicine system, an initiative. And what we are doing with the Singapore government is we're collaborating with the PRISM initiative, so their precision medicine initiative, with Sing Health and with Duke NUS. And the issues that they have in Singapore is, again, how to link the genome to the patients. So what we're doing is we have our technologies, our bioinformatics, machine learning algorithms that work in the background, and as we take data into the system, genomic data, we have images here of electronic health records, imaging, And a variety of data that is coming, but the microbiome data is just around the corner. Fitness and Fitbit trackers and all of that kind of technology is adding more and more data in. So when we collect this data, when the genomes are sequenced, the algorithms that we're running are delivering back, in the case of our app here, with pharmacogenomic information. So I don't actually often do this. Since giving up teaching live demonstrations, I haven't had to give very many of them. But I'm going to try one here now with the app that we've developed here. And so I've got it on my phone, and if I kind of start the app up, it will end up with the screen that you see here, just in case it doesn't work. Now, the genomic sequence that's behind the interface here is not my sequence. My sequence isn't ready yet. This belongs to our chairman and CEO. But if I click a little button here, it says, what is his drug response? So I will select warfarin, because it's a nice, easy one to type. And if I pop warfarin in here, it comes up with an image. It's either red, amber, or green, depending on how he pharmacologically responds to warfarin. And for some, it's an amber warning, which says, should he be prescribed warfarin, it should be prescribed in a low dose because he has a genotype, which means that drug will interfere with his kidney function. Now, I've taken warfarin for years. I don't have that phenotype, so I take it for a different thing. But if any drug that I go and discuss with my doctor is going to be prescribed for me, if we have that information, if we have that data, it's instantly accessible. Now, what was very good within Singapore is that individuals can come and interact, and they may see various different clinicians because they don't have a single GP, and this data is always available to all of them. And alongside the pharmacogenomic side of what we're doing with the app here, we have a number of other developments that we're putting in place with Singapore and some other sovereign entities that we're working together with on this project to put other important actionable gene categories. And they're all designed to actualize the potential of genomics, to be able to interact in a sensible way and in a joined up way with our genomic information. So the app itself is ready to use. It's being used, and it's in development. As I say, it allows patients to interact with the pharmacogenomics and their own profiles. We have interfaces for clinicians, so where it says reports and health data. That data is being put in by hospitals, being put in by clinicians over in Singapore, and it's dynamic. So it updates as information updates, and the information that we have concerning pharmacogenomics is constantly being updated as research goes ahead. So the way that this works, it will deliver dynamically insights as the situations change and as more research is available. And that push through notifications as well. So what does it allow to do? Well, it kind of allows a better targeting of prescribing of drugs. Research shows that if you have pharmacogenomic information, doctors follow prescribing according to that data. It also shows, research shows that if you as a patient have pharmacogenomic information, you're far more compliant with taking your dosing and drug regimes. And this all has a knock-on effect to kind of the costs of healthcare. And I was asked to talk a little bit about that. So with the pharmacogenomics, really what we can look at doing is targeting more effectively which drugs are being used with which patients. And if we take it on a global scale, we spend $1 trillion on drug purchase per year. And about 40 to 45% of those drugs don't actually have the desired effect for the patient that's taking them. And if we can target using this information, we can reduce that, and that's a vast, vast amount of money. So on a micro scale, it helps because you are not wasting money on drugs that aren't going to work for you. And on a macro scale, it works because you can target far more wider with budgeting. And I have an example of that with some of the research that we've done, the work that we have done in India. So the map of India here shows the 29 states. And the other thing that we do when we're gathering this information is we can look at the impact of pharmacogenomics on a much larger scale. So really what the map shows here, it's how people respond to a particular drug. And this drug is an immunosuppressant drug, and it's used for transplant patients as a patient has a transplant to stop rejection. And the way in which physiologically you respond to this drug, it is affected by a particular variant in your genome. So if you have one particular variant, you'll respond in one way to the drug, and if you have a different variant, you'll respond in a different way. Now, it's very important to get this right in the surgery and post-surgery. What we see here is that the map shows the frequency, so the amount of people that have a particular variant type. And we can see that there's pale and there's dark orange colours. If we could look on the kind of, as you look at it, the left side of the map here, that particular Indian state. So India has about 5,000 subpopulations, ethnic populations, and their genetics is slightly different between them. So the state on the left, there is a frequency of 11% of one particular variant. And the state across on the right-hand side with the orange, the frequency is 60%. So 60% response in one particular way to this drug in one state, and an 11% frequency in a different state. So the way that the Indian Health Service works is the states have their own budgets. So on one side, we've got a particular major response and an amount of a drug that will be required for the majority of the population. And on the other side of the country, it's completely different. So when they're kind of working out their budgeting requirements and the costs, this is just for one variant, one drug type. Using this kind of information, health administrations can kind of look at wider pictures within the populations that they are responsible for. And again, try to target and reduce wastage in what they're doing. So, really, I can't talk for any longer. I would like to. There's a lot to talk about, but I have to finish. So I'd like to say thank you. This work, there's about 55, 60 people in the company, a lot based over in India doing the work. In particular, Jonathan Picker, one of our chief scientific officers, thanks to him for his discussions with me, and also to Will, our bioinformatician, for the work that I showed you in India. Okay, thank you.
Thank you, Paul, for nicely pointing out the twin challenges of how we get this information back to the point of care, and particularly the GP level, which I think is a challenge for us, and how we integrate it with the other data sets. And I think there's a challenge here in Britain for us to think about how we do that beyond just a genetic, but getting the other data in the same ecology. Our last speaker, before we go to questions and discussion, is Vivienne Parry. I think Vivienne will be well known to many of you because she's a very well-known broadcaster. And we are quite privileged, very privileged, to have Vivienne as the Head of Engagement at Genomics England. Vivienne Parry.
Hello. So I thought I'd talk to you about pensions. Now I can see your faces as I say pensions, and no doubt your HR departments have tried to tell you about pensions. And I'm guessing you've been a bit resistant. I'm guessing that it's really hard for lots of you, and you have no idea, frankly, what a lot of them are talking about. But you do need some basics. For instance, you need a pension. You need to know that. You need to know the main types. And if you really wanted to know the detail, you'd want to know a trusted place where you could go and look up what a pension was all about. And you could wrap a wet towel around your head, and it would probably take you a while, but you'd grip it. And it's kind of just-in-time learning, isn't it? Oh my God, I know I ought to do something about pension. I'm 42. I need to know about pensions. Show me the site. Tell me what to do. I'll go and look it up now. You need to know what not to do. So that's a really important thing. So you need to know that you can't blow all your pension on a Ferrari. Oh, is that what you've done? You need to know as well that if a cold caller rings you up and says, "Madam, I think you should transfer your pension to my operation," you need to say no and put the phone down. And above all, you want clear language, because you don't want somebody confusing you with superannuation and contributory and all the rest of it. You just want the basic facts. Now transfer all that to genomics. And I started with pensions because lots of you know about genomics. And the tendency of all scientists is to think that everybody needs to know what you know. And they don't. It would be very nice, but they don't. They really, really don't. And you have to think, what is it that people really need to know about genomics? And I think the first thing is they've got to know what's in it for me and my family. What are the benefits? And I think that step is, what's in it for you is that this is the first step to personalised medicine for you and your family. Number one point. The second major point that I think that we need to get across to the public is a very simple message, and it's the one that Sally Davies so brilliantly articulated, is that it's about sharing. I'll get better results for me and my family if everybody else has shared their data. So it's not just I'm getting good results, but actually I'm relying on other people. And other people are relying on me to share my data. And in our post-Brexit days, when we sit in a huddle and we throw chair legs on the fire, there will be, I think, a need for us all to come together. And actually, for me, the thing about genomics that's incredibly powerful is that it harks back to the first principles of the NHS. We are all in this together. And actually, so if you get across that key point about sharing, I share, you share, we all get better care. It's a very important message to get across. And the third thing that people really need to know is about personal detriment. Because we did an enormous amount of public engagement at the beginning of the 100,000 Genomes Project about what people were really worried about. And the bottom line is, I don't want it to be harmed by this. I don't want to suffer financial detriment, so I don't want insurance companies to know. I don't want there to be somehow to be marketing. What was very interesting about the marketing, though, was that there were quite a lot of people who the most thing they were worried about was that somehow fluoromargarine would get hold of genomic data. And they would ring you just as you were sitting down to watching Downton Abbey on a Sunday evening, and they would say, we see you've got a high cholesterol variant. Have you thought about using fluoromargarine? So people were worried about the marketing, but it's a financial detriment in terms of mortgages, jobs, travel insurance, the whole range. That's what people are really concerned about. So a major piece of communication that people need to know is that their information is secure and confidential, and that it's not going to be used in a way that will be a personal detriment. So let's now come to the basics, because I think as I've gone through the journey with the 100,000 Genomes Project, I give out less information about DNA than I did in the past. So I actually did genetics at university. I was really bad at it. I mean, really bad. I was begged not to do practicals because I broke so much equipment. They said, we can't afford to teach you, Vivienne, please stop this. So, but there's a lot of stuff that we don't really need to know. So if I turn my microwave on, I don't need to know the physics of microwaves. I really don't. I just push the button. And I think there's a danger that we confuse with too much scientific information that isn't actually necessary. I think some of the basics that we do need to get across are you can have a simple test. It's just perhaps a single variant. Perhaps it's familial hypercholesterolemia. You can have a bit more complex, just related to your particular condition, a panel test. Or you can go the whole 9 yards and have whole genome sequencing. But you don't need to tell people about all the ins and outs of how it's done or what different sequences mean. That is not information that people want to be burdened with. On the other hand, they need to have the place that they can go where they can get really clear information if they want to be the nerdy one that goes and has to read it all. We need to provide that. And we have to provide it in incredibly clear language. So we did a big piece of work called Socializing the Genome, and we discovered that actually people know far more about genomics than you perhaps think they do. So although only 12% of people say they know about genomics, let me take you back to pensions. If you were jumped on by a man in the street with a clipboard who said, tell me about superannuated pensions, even if you had a superannuated pension, you'd go, and not answer and say you didn't know, simply because you thought that you would look a complete fool if you said you didn't know. So I think that actually when you talk to people, a lot of the principles behind genomics, they absolutely get and they understand. So I think the clarity of language, so one of our particular findings, for instance, was that people don't like you talking about mutations. They're not mutant ninja turtles. They really don't like talking about mutations. They would rather talk about a glitch. You've got a glitch. A glitch is kind of a quite friendly thing. A glitch is not really bad. It's might be quite friendly, but it's your glitch. And it's not a stigmatizing thing. So I think we have to be very careful about our use of language in genomics and the way we phrase things, and stop talking about mutations and stop talking about mining data or exploiting data. I beat people over the head at Genomics England if they talk about exploiting data. We don't exploit people's data. We use people's data. We allow people to have access to it because we want them to use it in a way that will benefit people. So let's go on a bit with that. So what else do you need to know? It's the impact on your family. Your test results may mean other people in your family need testing too. That's a simple message, but people need to know that what knowledge they get will also have knowledge for their family. And I think as we move on, I think that we come into a more difficult area. So DNA is only part of your health story. If I had an eating all the pies gene, if there were such a thing. It would depend on whether I had access to pies as to whether, that would be one of the things. I might not necessarily be as big as a huge pie if I ate all the pies, because if I lived on a desert island where there were no pies, I would never have an opportunity to realise what a pie would do to me. The pieman might have passed over my Tinder profile, and I would never get to meet the pie man and eat all his pies and remain slim all my life. But that thing about knowing the implications, because at the moment we're concentrating on variants that have a specific health impact, and that may lead people to believe genetic determinism, that every gene will cause something. And actually, as we go forward to a risk-based, there may be more of a story to tell there. And I'll finish there.
Thank you, Vivienne. Quite right. I think simple messages, people just want to know the take-home message for them and their families. So I'd like to invite the panellists to come up to the front. What we're going to do, I'm going to pose just a couple of questions to the panellists to get the ball rolling. While they're answering those questions, I'd love you to think about what you'd like to ask them, and I will come back and I'll take groups of questions so that we can deal with several things at once. And I'll start the ball rolling and just by posing the question to the panellists. Two questions. One is, how does genomics challenge traditional assessments and models of health economics? And the related question is that how does it impact or challenge traditional models of healthcare? So in no particular order, let's start with Sue. Would you like to pick up those, Sue?
In terms of your first question, it challenges the traditional models of health economics, because what it will enable us to do is to stop undertaking some of the investigations, or for patients to see different specialists within their healthcare system. The challenge is how we start to measure that impact and understand that impact. So there's no doubt about it that to look properly at how genomics is implemented in the healthcare system, we need to look at health economics and to understand the costs for the end-to-end patient pathway. I'm going to start on that question, and then do you want me to answer the other bit?
No, let's come back to the other one. We'll go up and down, I think. So, Nalin, would you like to?
I think Sue's covered it really well, but I think in terms of there may be cost at front end greater than cost, and there'll be cost savings later in the system, because quite a nice example you gave was early detection of cancer, for example. So the benefits will sort of feed through into the system, and you will recover. So the initial cost of actually doing the genomic studies implementation in the clinical setting, you will be able to recover from the benefits that are realized through the data.
Yes, I think the point to make is that whatever the reason for commissioning the analysis, you can mine it for lots of things, including adverse drug reactions.
Exactly.
So you get a lot more for your investment than a traditional test.
Exactly, yes.
Yeah. Paul?
Yeah, so I agree with everything that's been said. So if we have a genomic sequence, that genomic sequence is your genomic sequence, and as things progress with the research and the capabilities of interpreting that genomic sequence, if you actually have it in the first place, you can benefit dynamically from having that there. I mean, medicine, if I get sick, I see a doctor, the doctor tries to get me well again. If I have my genomic sequence, maybe we can turn things around and actually try and keep me well in the first place, and look at the actual cost savings that are involved, where I'm not actually going in saying, please make me well, but I have actually worked very hard to stay well. And if we look at end of life costs within the healthcare system, if we look at, I would like to live a very long life, but enjoy it, and then maybe have it end very suddenly, and reduce my morbidity. And if I can do that, I will save the NHS service an awful lot of money towards the end of my life. And actually genomics actually will facilitate in some way that reduction in the morbidity and the costs that are involved with that as well.
Thank you, Paul. Vivienne.
I think there's a thing about real-world evidence here, and the way that in the NHS traditionally we introduce new things, but we still keep on the old things, so that you have multiple systems going at the same time. And it goes back to education, really. So if I walked into my GP and said, you know, here are my genomic test results in relation to pharmacogenomics, my GP would probably say either, oh, you don't want to believe those companies. They probably wouldn't have a place to put it in the electronic record, and I probably wouldn't gain the economic benefits of having the right medicine. So I think that in the real world, as opposed to a kind of a research setting, we need to make sure that the education really delivers the step change in practice, which will deliver the step change in economic benefits.
Well, I think you've segued nicely into the change in practice. So, did you want to offer, Vivienne, any thoughts about what healthcare encounter for a typical patient might look like in 10 or 20 years' time? They're not sick to begin with.
It's funny because I used to do these sort of things like 20 years ago where we would say, oh, it's all going to change, you know, by Tuesday tea time, that was our normal time frame, you know, you will see people looking in and the great one I remember doing 20 years ago for television was you'll look in the mirror and your mirror will talk back to you and say, you're looking a bit peaky today. You know, I've looked at your genome, you haven't eaten enough sprouts recently, you need more B vitamins. I don't know, whatever. And I think that in some ways there's been an element of overhype for which probably I hold up my hand and say I was responsible as a journalist. That we need to say that actually that encounters are not going to change with doctors, in that doctors will still be seeing the patients. But maybe what I think we will see is that patients will be holding their own records. Because I cannot see that we are going to go on in the way that we are at the moment with records held centrally by hospitals and doctors.
When you're passing to Paul, I just recall Bill Gates' famous saying, at least I'm paraphrasing what he said, that new technologies are overhyped in the short term, but underestimated in the long term. And the reason for that is you need to put a lot in place before it takes off. And I think we're at that inflection point now. Paul, how do you see it going?
Well, I mean, I think it's been an interesting in the news over the past week or so with the way in which individuals are interacting with medical information, shall we say. So if I went in for an MRI scan back in June, I'd expect a letter to drop through my letterbox. Well, by now, I would have hoped, explaining what was going on. And when I read that letter, I think, well, okay, I need to go and see my GP because I need it explained to me, because I'm not sure exactly what it means, because I can't understand the language that's being used. And I understand that there's a big push to change the scientific language so that it's understandable by all and not just those with a medical degree. And I think as we move forward, we will be able to interact more with our own information, but in a way in which we understand, as Vivienne was saying just now, and actually having the ability to there's informed consent, but informed choice as well. Discuss with my clinician exactly what would be best for me, but in a way where it's not being, or not being told, or not being led, but I'm actually making an active choice. And to look at what those choices are for my treatment based on my information, and not based on, we always do this for everyone in your situation, and it works for the majority.
Yep. When I pass to Nalin, it reminds me of Eric Topol has a chapter on this in his famous book, and I recommend it to anybody who's interested in this area if you're not already familiar with it. The book is called The Patient Will See You Now, by Eric Topol's cardiologist in San Diego. And every chapter is an epiphany, but one is about patient empowerment, and it's very powerful. People will know more about their conditions and want a more interactive relationship with their healthcare professionals than they've had in the past, because there'll be much more personal data available. Nalin.
I think it's a mug's game trying to predict the future. I think if I just look at a smartphone, for example, and say what it couldn't do now, I could not have predicted even 5 years ago. So I think trying to predict what medicine or medical care will look like in 20 years is almost certainly not realistic. But
what impact do you think it will have in foreseeable future?
But I think there will be. It depends on how we integrate the information from our lifestyles and so on with genomic data. And I think wearable technology will have a big part to play in that. Information will empower us, but it can also have power to sort of imprison us as well. So I think it really, I don't know how it will play out, but I think hopefully it will be the positive that it empowers us to sort of make the right decisions, allowing us the choice to make the decisions we want, as opposed to being limited by medicalization.
Yeah. Well, I hope to go to my GP and get told that I'm B27 positive and I need this drug, not that one.
So, John, where we're going to be, there's no doubt about it, whether it's the genomic technologies that we've got available today, up to the level of whole genome sequencing, or the ones we anticipate coming into healthcare very soon. So that's looking at from DNA in terms of how it's transcribed, how it's then translated into the proteins that make us, into what type of things circulate in our body, that this is going to change rapidly. So genomics is going to be a disruptive innovation for healthcare. And I think that's pretty clear. That means that we will see new models of service provision evolving. We've already seen it actually as part of the 100,000 Genomes Project, and we'll just be taking that to another stage, which is trying to apply genomics in a coordinated way across the country, but in end-to-end patient pathways. And what this is going to drive is that we're going to move from what was protocolized care, that moved into evidence-based care, that's now going to move us into data-driven care, and large amounts of data that's going to drive the way in which we both deliver services, but in the way in which patients can receive either predictive risk profiles or more precise diagnosis or more personalised treatment. This will be all encumbered on us, one, making sure that we continue and we can fund the infrastructure and we can fund the changes, but we can take out the technology that's no longer required to make this affordable within the system. And what we all know, and I think those people who've been leading and speaking on the genomics revolution for a long time, we know this is going to fundamentally change the way that medicine's practiced and the way we characterise diseases, away from individual specialties into something that might be, whether it's an immune function problem, that might be an immune function problem that affects the lung, or it could be affect the joint. So we're going to think about things very differently.
Yes, we will indeed. All right, well, I'd like to open it up to everybody in the audience to ask our panel questions. Great, we've got some hands up already.
First, I'd like to thank all the speakers for coming and speak to us at this time of the day, and learned a lot. Thank you very much. So I have two questions. So first, I agree that genomic data is definitely the fundamental and the most important data for future healthcare and medical kind of research and more like day-to-day delivery. But I'm just wondering, like for individual diagnostic point of view, is there going to be like further like drive into, for instance, epigenomic data, transcriptomic data, and proteomic data, because for individuals, they are going to be more accurate to predict how a patient is going to respond to a special treatment. And my second question is how this push into the genomic world will, in your view, would drive the changes to future healthcare industry, for instance, pharmaceuticals. Would you think that this increase cost of looking into real-world evidence and personalized medicine would drive further disruptions, or actually make it harder, create higher barriers for the new players to join the industry? Thank you.
Hi, thank you very much for the talk. I just have a question for you, Ms. Suhail. How do you see the NHS, what role the NHS is going to play to bring a change within NICE guidelines? Because as you know, to have a change that is crystallized and kind of materialized, practitioners usually follow specific guidelines as to their clinical practice. And usually these guidelines are the ones that are published by NICE. So how is the NHS going to play a role in bringing that change? Thank you.
Well, perhaps we'll get Sue to actually elaborate what the change might be required at the NICE guidelines to cope with genomics.
Hello, my name is Nana. I'm a trainee bioinformatician and very fortunate to be part of the one of the genomic medicine centres that's going to be delivering whole genome sequencing this year. My question for the panel is that I think that's a really good question you posed there, and I think that there's a lot of companies outside of health that are making genomics everybody's business. So people see the value in things like ancestry information or finding out their genetic risk about certain diseases, and I just wondered whether healthcare and the NHS had any view on that conversation that's going on. Thank you.
So, 2 or 3, 4 really interesting questions there. Sue, would you pick it up and then we'll just move along and address any of those questions that you feel?
First of all, how are we going to encompass other, I refer to it as the other components of the functional genomics pathway, so transcriptomics or epigenetics or proteomics. The National Test Directory that will drive our genomics testing service, that's for rare disease, as well as for some common diseases, as well as for cancer, for pharmacogenomics as well. What we've already set out is that as the evidence emerges for using transcriptomics, for example, then we will consider that in terms of how that should be included and to roll that out systematically. And we already know from the work today, we launched the Public Health Genomics report into emergent technologies, and we know transcriptomics and circulating DNA are 2 areas that we're going to need to look at. So this test directory and the evidence that's available to support the introduction of new tests will be reviewed annually, and therefore that will drive what is provided within the NHS. The second question, which wasn't asked by you, but it's around NICE, I think there are a couple of things here. One, historically, NICE have not recommended the genetic tests that have been undertaken in the NHS. This has been done for rare and inherited diseases, mainly by the UK Genetic Testing Network. That's now come into NHS England, and a new system will be introduced. But there is the question, I think, about genomic tests that are used as a companion diagnostic for treatment entities, which is often the case in cancer, and how NICE might start to consider technologies that look potentially across different types of cancer, and how that might be recognised as a companion diagnostic for not just a single treatment agent, but perhaps for many. But the conversation we had earlier today as part of the PHG report launch was, in terms of the evidence that might drive the diagnostics, we need to start to think about real-world data and real-world evidence and consensual data rather than grade A data. So I think there's a lot of conversations that we need to have. Shall I stop at just answering those two, John, or do you want me to continue?
No, I'd stop at those two and come back if you see that all of the questions haven't been answered, because I'd like to give all the panelists a chance. Nalin.
If I can address the issue about using epigenomics and proteomics and so on. I mean, DNA is a very robust molecule. RNA, proteins less so, and they're very labile. And the technology is not at the stage yet. It is certainly with genomics analysis for us to be able to sort of use it robustly in clinical practice. So there is a lot of work that still has to go into this until it becomes usable. No doubt it will, and we are seeing in a research basis very interesting results, but it needs to be robust enough technology to be able to use in a clinical setting, and we're not at that point yet. So I think it will happen in time, and yes, you will need to integrate that information together with genomics information, together with lifestyle information, all of those things to get a more accurate prediction.
Cool.
So if we think on the commercial company side of things, and how is genomics, and looking at that question, if we think of drug discovery, actually speeding up failure in drug discovery is a very good thing. If we look at how many potential drugs actually reach market compared to fall by the wayside, I mean, there are vast quantities of money every year that are poured away by these companies as things fail. So if we can use genomics to help speed up drug discovery, but also speed up the failure of drug discovery, and that's a very important point. And if we have a kind of shift in the amount of money that's being wasted in that R&D, and we couple that with a shift in the amount of money, as I said when I spoke, of drugs that are prescribed for indications. If we look at different indications and the percentage of drugs that fail to initiate any response in patients, on a global scale, if we step out a bit further, it's a vast amount of money. And the question is, what would we do with that money if we saved it? And if we were to put that into healthcare.
Paul, I might pick up on that point just to point out to the audience that many drugs fail clinical trials at phase 2 because, particularly cancer drugs, 5 or 10% of people typically will respond brilliantly, but the rest won't. And that's not a high enough success rate to go into the very expensive phase 3 trials. But if you can work out why that 5 or 10% has responded so well, you can repeat the phase 2 trial on a subset of the people that have that genetic variation, get a high success rate, which means that phase 3 trials will be much shorter and much cheaper as well, because phase 3 trials get finished or terminated when there's sufficient evidence. So I think it's going to change a lot of the dynamics. Many drugs that are useful for you and me, but not for most people in the room, will be rescued. And the whole cost structure, including the very expensive cost structure of these on-patent drugs, will change, because you won't be spending quite so much money or nearly so much money on very expensive trials on those that work well on the average and losing those that will work well on just a percentage of us. So that's an important point to make. Vivienne.
So can I just come back to making the companies out there that are already, as you said, making genomics everybody's business. I mean, companies like 23andMe, and every day I get stuff in my inbox which tells me that there is a new test. I mean, I've been off myself for a gene test in an incredibly swanky salon in Bond Street. To test whether I was suitable for one kind of wrinkle care or another. And the creams were, by the way, £600 for a month's supply. So there is an awful lot of complete nonsense out there in terms of genome information. And it goes back to the point I was making earlier about people being told that there is a gene for this and a gene for that. There's a gene for wrinkles and whatever. And it's kind of misleading people and taking them down a road which actually isn't very helpful because it leads them down this road of genetic determinism. And also there is some right old nonsense in the ancestry field. So if you think about it, if you're the product of your parents and then your grandparents, and then it's doubling and doubling and doubling, actually, you very soon, quickly, as far as you go back, exceed the population of Britain. And I think the singularity comes around the 10th century. So if you're white and British, you are absolutely descended from the Vikings. We all are, because that's the way it's... But there's a lot of people making a lot of money out of it, and I think it's rather muddying the waters. However, on the other hand, 23andMe have the most fantastic patient information. It's absolutely wonderful. They've put a real amount of thought and co-production into that information. And I think we should steal from their books about the kind of information they're using, but actually try and rein it back in terms of what we're actually promising genomic testing will deliver for people.
Thank you, Vivienne. Sue, did you want to supplement any of those answers?
I think just a couple of points, John. The first is what we have to do going forward is recognise we may need to take into the healthcare system genomic information that might have been generated by people accessing companies outside of the NHS, and we have to have a way of assessing whether that information is good, bad or indifferent, as Viv has said. But equally, I think we need to open the conversation, and you and I have had this conversation, John, about how we use research data, research genomic data, to inform a patient's care, because that isn't necessarily always the case in many of the big research studies. So we need to ensure that all the genomic data that is generated really can be used to benefit patients. I think the second point that I wanted to make was what we also have to do in thinking about what we're trying to create, and this being everybody's business, is how can we also create a very vibrant ecosystem for all the companies in the genomic space, with the opportunity to test, their products through an analytical pipeline, so that we can really start to bring the benefits of new technologies into healthcare more quickly. And that's going to be important not only for the country, but also important for the patients at the end of the day. So we in NHS England have thought about that, working in partnership with Genomics England, but there's a lot more that we need to do to make sure that we benefit, or the patients benefit, from the very best possible technologies for sorting out their disease or giving them predictive risk profiles.
I think one of the things that becomes obvious when you think about it is that each national jurisdiction, given the different legal frameworks, will, I think, have to develop a central source of evidence-based, well-curated, continuously updated information on genomic variation that can be reliably put into the clinic, almost like having an FDA for that. And I think that then creates the raw material for companies like Paul's and others to package that up and bring it out to both patients and to physicians. But I do think that in Britain here we do need that central source of reliable information about what a genomic variation might mean and the predictive limits that one can put around it. So, right, well that was not a bad start, but it's a bit like being in church. I think we need a little more agitation here, so let's have some more questions and make them as fun as you can. There's one at the back.
Mark Rapa, I'm teaching assistant at the University of Manchester. First, I want to make a comment. I really liked the analogy with the pension. I come from a background of HIV activism, and what I've learned in the last 4 years of HIV activism is that the patient, there will be several occasions where the patient knows more than the clinician. Yes. So it is very important not to underestimate what the patient knows and what the patient expects from a treatment or from a counseling session. Secondly, it's probably the elephant in the room. Do you think in any way Brexit is going to affect the project, especially in line with the new clinical trials regulation, data protection regulation. Do you think in any way Brexit is going to affect what is happening with the project at the moment?
I'm John Aplin and I'm a professor here in the medical school. We're heading rapidly to being a two-tier society, those who've been sequenced perhaps because they've had cancer or a rare disease, and those who haven't. And I wonder what the vision is for sequencing 55 million people in a way that will give them equal opportunities to benefit from pharmacogenomics and better prognostics, etc.
It's a very good question. I'll give a partial answer because I've thought a lot about it. So the quick answer, but I'll pass the panel for more a fulsome response, is that in going from 100,000 to 55 million, you want a mix of strategic and maximum health value. So try to identify those subpopulations that are going to benefit early from doing this. But have a program, and I think we are discussing this about replacing the Guthrie test at birth as one way of democratising it, because you're not going to do it overnight. But so that might partly answer your question. Lady over here.
My name's Debra Bloor and I work in fertility.
You work where, sorry?
In Manchester. I work at a fertility hospital in South Manchester, but have been a regulator in the past and in fact worked with John. My question is at the other end really, so quite rightly we're focusing very much on us as citizens and patients owning our own genomic data, but going back to some of the questions we've had about NICE, and we've talked about the impact that the genomic information will have on potentially clinical trials that our big pharmaceutical companies will be doing, so they'll be more targeted and they'll be able to show greater effect, hopefully, of drugs. Are we ready, and is the organisation ready, to share this information with pharmaceutical companies who will make money from it? Are we as citizens ready for that? Because I quite would like my genomic data to be shared with AstraZeneca, thank you very much, because hopefully they'll develop a new drug that will help me if I get sick. But I think that's a big question and people will be suspicious of that.
Well, I'll pass over, but I would observe that without the drug companies you wouldn't have any drugs, and they invest in the clinical trials in the post-thalidomide era to make sure they're safe and effective. So it's a necessary evil, and I don't think they're evil. I think that companies need to be regulated, but that's about it. We'll start there. Vivienne, how do you see the public's response to sharing data with pharmaceutical companies to make new drugs?
It's very interesting. So people are suspicious. And they're suspicious because they think that, well, first of all, they misunderstand their place in the cycle. So they think that it's their data that has directly, it's like winning a lottery ticket. You know, you've got those numbers, so therefore, you know, the $64 million prize it is yours because you put your data in it. So they don't see that it's an enormous amount of data plus an enormous amount of effort by the company in order to take that forward. But they're suspicious about industry because they think that the medicines that they have contributed their data to may not be available in the NHS. And I think that's a major concern. In other words, that money will be made in other territories and somehow the NHS won't be able to afford the outcomes of the research that's done here. So I think that, George Freeman was very clear about this when we originally started the project, that actually the NHS would benefit in some way from whatever discoveries were made. I mean, each would have to be a case-by-case consideration, but the NHS would benefit in some way. And I think it's very important that we're upfront with people about how the benefits are going to be shared. I don't think people have a problem with profit from industry. What they have a problem with is disproportionate benefit. In other words, the benefit accrues all on one side and none on the other. So I think we have to put the mechanisms in place to make sure that there is a more proportionate benefit sharing.
Very good. Paul? What's your perspective
Okay,
on
so
any of the questions?
if we come to the question on the far side about the two-tier and genomes, I mean, we can look at this from a couple of aspects, but one aspect is the actual cost of genome sequencing. I mean, genome sequencing costs have tumbled dramatically since the Human Genome Project. For the first genome, £2.7 billion in 13 years. Today, $1,000, £900, £600 if you know the right people. But if we can bring that cost down to $100, under $100 per genome, that'll facilitate, as you're saying, a much wider genome sequencing program for a much wider population. So there's changes in technology that will go hand in hand with changes in the way in which the genomic sequencing is offered. And it'd be great, as you say, with the kind of the neonatal screening as a starting point. But with the whole genome sequence, the cost of that, in many cases, is offset by the costs of different either treatments or tests to try to find out if you're looking at a diagnostic odyssey, for example. So for rare disease, there's various different research, but if it takes 5 to 7 years to diagnose a rare disease with a slew of different tests, that time is reduced potentially with genomic sequence as well. So there is, I think, with technology changes, hand in hand with just changes in the way that the genomes are used, things will even out on that front. And if I have time, so with the idea of consenting and using genomic information and allowing others to use your genome information, I think, I agree that you should have the ability to allow others to use your genome in the way that you want. If you know what that means, and I don't mean that, certainly don't mean that in a patronising way. I mean, people need to understand the implications of consenting and allowing their genomic information to be used, whether it's in a clinical setting, a research setting, in a commercial setting. I mean, there's a real value in using the genomic information. And medical advances won't happen as quickly without it. But as long as you know kind of the who, the what, the when, and the how, I think you should be able to make the informed choice. So who are you allowing to use your data? A doctor, a clinical lab, pharmaceutical, an insurance company, a different commercial organisation. I mean, what are you allowing them to have? Are you allowing them to have your whole genome? Are you going to give all of it, part of it? You could give it de-identified, you could give it identified. When? Are you going to give it to them in perpetuity for a small amount of time, for one bit of research, specific study? And how are you going to give it to them indirectly, directly, via others' oversight? So if you have that informed process and pathway, then if you want to give your genome for everyone, for all, you can. If you want to do different things with it, and having the ability to choose what and who and where and how, I think that needs to be in place, and things will progress very quickly.
There's a number of business models around that very idea emerging. Nalin, what do you think?
If I can address the Brexit question.
Yeah, right, good.
I don't think it will make a serious difference, because the moves by MHRA, HRA are all towards keeping our process harmonized with Europe. There is goodwill in Europe, despite the politics, to sort of work with us, so I don't think it will change. It may offer us distinct advantages. I'm not a leaver, but our processes are quite aligned to them, but it may, after Brexit, we may have some flexibility, so some types of research we may not sort of go through. We may have a much more expedited, simpler processes, which at the moment we don't, because we have one standard process. So it may give us some flexibility, which will actually help us facilitate research a bit more. In terms of what John asked, in terms of having two-tier system, I think you will reach an inflection point where the benefits of genomic testing are so widespread and not limited to the groups that you currently sort of work on, that it will then become combined with the ever-reducing cost of genomic sequencing, will sort of make it viable for a wider population to be able to benefit
Yes,
from
the cost is still, I'll come to you in a second, Vivienne, the cost is still a factor. But you're quite right, as the costs come down, it's going to be easy to democratise it.
Yeah.
At this point, and the hardheads in, NHS and Treasury, etc., want to know what the return on investment might be. So we're looking to see where the maximum health value is at this point. Understanding, or some of us, that the real health value is when everybody is sequenced, because we'll be able to project forward for those people.
But it's what your very first question about how it'll change the health economics.
Yes.
When we reach a stage where the benefits outweigh any cost,
Yeah.
and it provides a cost benefit,
And
then that will be the sort of
yes, and we're probably not far away from that, but we need more competition in the market to drive the cost down. Vivienne, you were keen
just
to
a
add.
quick comment about Brexit, which is about bioinformaticians. Where's our bioinformatics guy, who was at the back, I think? So, by the way, if you want your children to keep you in your old age, get them to go into bioinformatics, because there's fantastic jobs, and they earn squillions because there aren't enough of them. And that's the issue, is that in Genomics England, for instance, I can't remember exactly what proportion of our bioinformaticians are from European Union countries, but it's quite a lot. And that would be a real disaster if all the bioinformaticians who currently work so hard to deliver all of this suddenly decide to go back to their home EU countries because they're not feeling welcome and their situations are uncertain. I think that would be a dreadful disaster.
Well, speaking as a foreigner, I feel very welcome here, and I'm not even in the EU.
Good.
Sue, please.
I think, though, Viv, part of what we've got to do is make sure that in all of the work that Health Education England do, that they recognise the importance of some of the specialist skills that we're going to need in the NHS. And if we can do this together with other industries as well, we'll make sure that we'll have a thriving education and training programme for those individuals. And that's what we're trying to do. John, very quickly wanted to come back on a couple of the points. First of all, the issue about the relationship with the pharmaceutical companies. And I think as we move forward, we have to get into a new relationship with them for the very reason that you outlined, that the efficiency of their drugs will be reduced when you start to genomically characterise patients. So their potential market will be smaller for some of their agents. But part of this is how do we use real-world evidence to drive a new relationship with the pharmaceutical companies so that they don't actually price the NHS out of the market and them not being, therefore, those drugs being available. And we're very aware of that and are going to work through that in terms of how we can address it, because it's the quid pro quo on all of this. The second is the equitable access. There's a question about, we've had the discussion about whole genome sequencing, and the cost point is just too prohibitive at the moment. But in terms of what will be available through the NHS genomic test directory, there will be access that's across the care continuum from primary care through to secondary and tertiary care to many genomic tests that have not been available. So, for example, we do very little testing for familial hypercholesteremia, so to look at the variants associated with high circulating fat levels. We are starting to address that, although that will be in more running panels of genes in that area. We're already starting to look at how we would move forward with pharmacogenomic profiling, particularly associated with adverse drug reactions, which we've already heard about today and cost the NHS an awful lot of money. So there are things that we can do today that will actually improve outcomes for people without moving to whole genome sequencing, and we have to make sure there's equitable access to that. I think the final point is where we are with the 100,000 Genomes Project is we haven't started to receive the incidental secondary findings or whatever you want to refer to them as from whole genome sequencing yet. So we're not able to evaluate the end-to-end, both value of this to the individual, but also the cost to the service for implementing it. And as we start to think about rolling anything out, if the price point was right, we'd have to do that end-to-end evaluation to enable us to introduce that in a cost-effective way within the health service.
Thank you, Sue. Now we've got 7 or 8 minutes left. Maybe time for 1 or 2. There's 2 I can spot. The lady in the corner, we'll go with her first, and the gentleman on the left up here. Bear in mind we've got 8 minutes.
Good evening. I'm really sorry to disappoint you, Vivienne. I'm another bioinformatician, and we don't get paid as much as you think. If I do go into bioinformatics is one of the lowest paid IT jobs. If I do go into financing sector, I'll probably get paid twice, but then we would have to sell the soul. So anyway, on a serious note, my question is that in India, public healthcare and genomic data awareness is very different compared to UK. Privacy, consent, regulatory affairs, and standards are not yet in place, especially in this area. This could mean that genomic information and pharmacogenomic data can be misused for profits. So how is Global Gene Corporation dealing with ensuring that this doesn't happen?
Right, so that's a particular question for Paul.
Hello, my name is Keith Hart. I'm a lecturer in biomedical sciences from Cardiff. I'd just like to ask the panel, as well as the chair, if they've had their own genome sequenced. If not, why not? And are they prepared to share anything about what they found?
Well, I'll start. Well, I have. Anybody else? Right, so we're 3 of us. Quick story on my genome sequence. As I told my wife, I'm almost perfect, but I do have 1 or 2 glitches. I think that's a good term, Vivienne. One of which is I'm a carrier for cystic fibrosis. And I never knew. There's no family history. I've got the Irish version. I blame my mother, but I can't dig her up to check. She was Mary Brady was her name, Irish. But the reason I bring it up is because it tells you that family history is not very useful. This is one of the most common genetic conditions in the population. 1 in 20 people of Caucasian origin have that allele. And so even in my extended family, it hadn't shown up. My wife is largely Irish in ancestry, and she has a 1 in 20 chance of being a carrier as well. And I don't know if we dodged a bullet with our kids or not. So I think having that information in the future will inform preconception advice, and I think will radically reduce the incidence of disabled children being born. Because if we're going around the block again, we're not. But I'd insist that she be tested, and if we really want to have another child, I'd go straight to IVF, which is an invasive and difficult procedure, but it's a lot better than having a disabled child. That's my story. Sue, do you have one from yours?
I do. I'm a participant in the 100,000 Genomes Project. When I started leading the NHS contribution to the 100,000 Genomes Project, I didn't ever think that I would be. But last end of November, early December, I was diagnosed with breast cancer. I'm still in the middle of active treatment, so I'm still receiving radiotherapy at the moment, but I've been through 9 months of treatment. And at surgery in January, my tumour and also my blood was sequenced. So what I know is I have a genomically quiet tumour, and it didn't reveal much more than the standard testing in the NHS. But there is one particular mutation where some of the newer drugs might be important for the future. Why it's been important? Well, one, of course, I'm passionate about genomics. I'm passionate about the 100,000 Genomes Project. I'm passionate about bringing genomics into healthcare and to be better understood in terms of the management of cancer. And although that might sound a strange comment, but there's more work we've got to do. But for me, what it means is further down the line, if I do get a recurrence, then I've got a very good baseline on which to understand what other treatments that I've got. But unfortunately, it's not very pretty in terms of its genomic profile.
Vivienne might have something prettier. Perhaps a wrinkly gene.
So I've had some it's kind of nonsense consumer testing, wrinkles obviously. I'm really 85. And not terribly helpful. All those things about fitness, you know, I'm built for endurance. I'm built for endurance, but the bad news is that the joints are going to be knackered. I mean, so there's lots of stuff which is really a bit confusing and contradictory, and do I set a great deal of store by it? Well, it's kind of like horoscopes in some ways. You know, do you ever read your horoscope? You've kind of got a buffer memory, like a printer, haven't you, for a horoscope? You read it, you retain it for about 5 seconds, and then it's gone. And I think some of the consumer testing that I've had for genomics has kind of stayed that long.
Yeah, but that's the pop stuff. We're talking the serious end here.
This is the pop stuff. But would I go along to 23andMe and ask for genomic testing for, say, Alzheimer's? No, I'm not sure that I would, and certainly not yet. I mean, I'd want to know, I guess like a lot of people, that there's something that active that you can do. Knowing that there's something that you can't really do much about isn't, I think, that helpful. Would I choose it to choose my mate in life? Luckily, I've found the perfect man already, but because there's quite a lot of that, genomics for dating.
Is there?
Oh, yes.
Yeah.
Oh, yes, there's a lot of genomics for dating, and a magazine did ask me to suss it out, but I refused that assignment.
Thank you, Vivienne. I hesitate to throw to Paul, but because I'm hoping we can finish on a high note rather than a low one. But there was a question about the reliability of information in your jurisdictions like India.
Yeah, so to answer the question, obviously, GGC, we've been working in India since we started. And yes, it's a very different situation, but we have built India's largest consented biobank. And that consent means that the participants that we have in our biobank are very much informed of exactly what's going on. We don't pay participants to join our biobank, and it's done in a very open manner. Our headquarters, the company headquarters, is in Singapore. Singapore has a very rigid and very clear ethical guidelines in this area, and we follow those implicitly. So, I don't think we would be able to do business if we were doing bad business.
I think that's true all over the world now. I'd like to finish now. We are on the verge of a new era where what's under our hoods can be taken into account in the way our healthcare is managed, why some of us get asymmetric arthritis and some don't, for example, and what's the right treatment for that. But it has to be done responsibly, and I think that's the important role that Genomics England and NHS play in making sure that the benefits of genomic information are brought responsibly but actively to the clinic for the benefit of the patients. I'd like to pass back to Sarah now to close off. Thank you, Sarah.
Before we thank tonight's speakers, we need your help. We're a small charity, as I said at the beginning, and we need your help to keep going. So anything that you can do to support us, it'd be much appreciated. So become a friend of PET, give us a regular donation every month. That's fantastic. Just £3 a month, so a cup of coffee. I'd really like to thank our partners, Genomics England, in helping put this event together, to Kat Nevin Ridley and her team. And it's a little bit of sadness because this will be the last event that we're working with Kat on, and wish her good luck in her new job. And I also want to draw attention to the information in your packs about our next events. Our next event in the Genomics Conversation is going to be taking place in Edinburgh, and that's called Whose Genome Is It Anyway? Big Data and Your DNA. So there'll be lots to talk about that. And also I'd like to draw attention to our annual conference, which is in December, and that's called Make, Do or Amend, Should We Update UK Fertility and Embryo Law? So I'd like to thank my team who've helped put the event together this evening as well. And I'd really like now to thank the speakers for sharing their expertise. Sue, Nalin, Paul and Vivienne, thank you so much for all your contributions. And I'd also like to thank John, our chair. We hope you've enjoyed your first trip to Manchester, and it's great to know that you're almost perfect. But really, I love it when a chair shares their expertise and joins in the discussion and enriches it in the way that you did. So thank you so much for that. So thank you and have a safe journey home.